So today let us know the chemistry of Maxam Gilbert sequencing. Today we will mainly focus on the most fascinating aspect of Maxam Gilbert sequencing, the chemistry that makes DNA sequencing possible. So unlike modern sequencing methods that rely heavily on enzymes, Maxam Gilbert sequencing uses a series of carefully chosen chemicals to identify the positions of individual DNA bases. Imagine that we have a DNA molecule whose sequence is unknown. The first step is to label one end of the DNA with a radioactive marker so that we can later visualize the fragment that is produced during the experiments. Once the DNA is labeled, it is divided equally into four separate reaction tubes. Each tube contains different chemical environment which is designed to target specific bases within the DNA molecule. In the first tube, which is guanine or G reaction, we add dimethyl sulfate, commonly known as DMS. And this DMS is a methylating agent that specifically attacks guanine residues, adding a methyl group to the N7 position of guanine. So you can think of DMS as placing the tiny chemical flag on every guanine in the DNA strand. At this stage, the DNA is not just at this stage DNA is not cut actually. The guanine bases are simply marked for identification. The second tube is the adenine plus guanine or A plus G reaction. Here what happens is the formic acid is used. So this formic acid acts on the purine bases adenine and guanine, weakening their attachment to the sugar phosphate backbone. In essence, formic acid prepares both the adenine and guanine sites for later cleavage. And this reaction allows us to identify purine positions within the DNA sequence. The third tube contains hydrogen along with a high concentration of sodium chloride. Hydrogen naturally reacts with both cytosine and thymine, but the presence of sodium chloride suppresses its reaction with thymine. As a result, only cytosine residues are modified. So this is a clever chemical trick because the same reagent can be made selective simply by changing the reaction conditions. In the fourth tube, hydrogen is used again, but this time without sodium chloride. Without the salt, hydrogen reacts freely with both cytosine and thymine. So consequently, all cytosine and thymine residues become chemically modified. Comparing the results from C tube and C plus T tube later allows us to distinguish between these two pyrimidine bases. At this point, each reaction tube contains DNA molecule with chemically modified bases. But the DNA strands are still intact. The next step introduces the most important reagent in the entire procedure, hot piperidine. So this piperidine is added to every reaction mixture and it recognizes the chemically modified bases, removes them, and breaks the sugar phosphate backbone at those positions. In other words, piperidine converts chemical modification into actual cuts in the DNA strand. As a result, each tube now contains a collection of DNA fragments of different length. The G tube contains fragments ending at guanine positions. The A plus G tube contains fragments ending at adenine and guanine positions. The C tube contains fragments ending at cytosine positions. And the C plus T tube contains fragments ending at both cytosine and thymine positions. So these fragments are then separated on a high resolution polyacrylamide gel. Smaller fragments travel farther down the gel while larger fragments remain closer to the top. After electrophoresis, the gel is exposed to X-ray film, producing an autoradiograph in which each DNA fragment appears as dark band. To determine the DNA sequence, we read the band from bottom of the gel upward. The smallest fragment corresponds to the first nucleotide from the labeled end, and by observing which lane contains each band, we can identify the corresponding base. A band with G lane indicates guanine. A band in A plus G lane but not in the G lane indicates adenine. A band appearing in both C and C plus T lane indicates cytosine, while the band appearing only in the C plus T lane indicates thymine. Thus, through the combined action of DMS, formic acid, hydrogen, sodium chloride, and piperidine, scientists are able to convert an unknown DNA molecule into a readable pattern of bands. The brilliance of Maxam Gilbert sequencing lies in this elegant use of chemical specificity. Each reagent performs a unique task, and together they reveal the genetic code hidden within the DNA molecule. This method stands as one of the class greatest achievement of modern molecular biology, demonstrating how the principles of chemistry can be used to decipher the language of